and welcome to the latest Spotlight On interview. Today, I'm joined by Karoline Kuchenbaecker, Professor of Genetic Epidemiology at UCL. She's going to talk to us about her research into the genetics of complex traits and the importance of increasing diversity in genetics studies. So, without further ado, Karoline, could you introduce yourself and perhaps give us an overview of your work?
Hi, Lauren. Thanks a lot. I am a genomics researcher at UCL, as you mentioned. I've been doing research about the genetic causes of diseases for quite a long time, and I've actually worked on different diseases over time. I still do it. The main focus, the one thing that connects all of my work is questions around diversity, especially people of different ancestry, and whether that matters and how it could impact findings in genomics, and especially when we think about genomic applications for medicine.
Brilliant, thank you for that. So I'd love to know how you first got into this line of work.
Actually, that's a little bit of an anecdote. I mean, I got into genomics in general just because I thought it was really interesting and so new. And I studied psychology previously and statistics, but they're all relatively older fields. And genomics was so exciting because you could find new things. But this diversity question emerged literally on one particular day when I was teaching in Cambridge. And I discussed how we do these genetic studies. There was a student at some point who was really having a huge impact on me. I was explaining how we're looking at ancestry in these studies, and then usually we try to use genomics to infer the ancestry a person has, and then we usually exclude every single person who has any non-European ancestry. I just really very routinely went through this, and that's how genetic studies were done then and are still done today. And that student I mentioned, who I'd say herself was not white, raised her hand, was completely shocked and just asked, why? So why are you excluding everybody? And it hadn't occurred to me, if I'm completely honest, that that's, kind of the massive implications of doing this and what it means to somebody who's maybe not self-identifying as white European.
Yeah, I mean, that's a great anecdote. And obviously we'll get into that a little bit more in some of the later questions. But I also wanted to ask you, so you're scientific lead for diverse data at Genomics England. Could you tell us a bit about what that role involves?
Yes, sure. That's actually a great new initiative at Genomics England. Genomics England, just for a bit of background, is a government-owned company that's overseen a massive sequencing project in the UK, the 100,000 Genomes Project. And that's been completed, and they were really, the Genomics England is still a key player in the very clinical genomic medicine implementation, so how the NHS is using whole genome sequencing. But more recently, there were new ideas for really massive projects. One very exciting initiative is the newborn screening project, and the other one is dedicated to diverse data. And as you can imagine, that's the one where I contribute to. So there's significant funding to look at diversity and improve things really from different perspectives, and make sure that genomic medicine in the UK is as fair and equitable as possible.
Yeah, that sounds like an amazing project. Perhaps I'll have some time to ask you about that later on. But yeah, so obviously a big focus of your work is on filling this gap in the diversity of genomics data. And I know you recently published a sort of roadmap on how to fix this in Nature. So before we delve into that a little bit more and like sort of some of your recommendations around that, could you perhaps give us a bit of context? So how diverse are current genetic studies? And is this a trend that's improving, or how is that going?
Yeah, important question. Currently, genomic studies are not very diverse. Around 80% or so of published studies use data from people of European ancestry, so the vast majority. And if you compare that to the proportions of world population, obviously not 80% of the world population are white. For other major ancestry groups, there's also a big imbalance. So the second largest groups, people of East Asian ancestry, and some groups are really, really severely underrepresented. The thing that really surprised me was when we looked at these numbers in that roadmap paper you mentioned. We looked at it over time, and it hasn't really changed. So things haven't improved in terms of proportions in the last years or so, despite repeated calls and despite a lot more awareness compared to the days when I started out in genomic research.
Yeah, I remember reading the same thing in your editorial, and it was also a massive surprise for me. I had assumed that it might have improved, especially over the last few years, but like you say, showed that actually the trend, if anything, is going down. So yeah, I must be sort of frustrating. But I guess as well, I wanted to go sort of back to the basics. So this might seem like quite a basic question, but why is it so important to include different populations in genetic research?
Yeah, I don't think it's a basic question at all. I think it's actually there's ongoing research to really pin this down. So fundamentally, I mean, all people are genetically, very, very similar, and the differences between different ancestry groups are minor. But by focusing on predominantly one group, we're still excluding a lot of genomic diversity. So we're not really having the full picture. And there's a lot of discoveries we're missing out on because there's so many people we're not including. Then there's also some practical consequences. So for example, if we're systematically not studying people of African descent or from certain regions, there may be particular mutations in those groups that we're not actually aware of. And then if we do genomic screening in the NHS, we may be missing really important mutations a person may have just because we haven't, or we might just not be sure if this is really, impacting on the disease or not. And that's really problematic for people. And the third very important bit is predicting risk based on genetics. It's been really well established that that doesn't work very well across different ancestry groups. So as we do most research in white people, predicting risk works by far best in white people.
Yeah, which is obviously a massive problem, for healthcare generally as well. Yeah, I think as well, I mean, the main takeaway from that Nature editorial that you published was as well that it's not just important for those populations that are currently being missed, but actually for everyone. And like that's a massive part of this work that you're doing. So I wanted to ask you as well about what sort of factors have contributed to underrepresented populations being absent from research. So could you perhaps explain some of the reasons why there is that current lack of representation?
It's clearly a really complex story.
Yeah.
From the probably quite naive perspective of the researcher, or having been myself part of that sort of more mainstream genomics research group, the reason we gave and the reason we was believed in was that if you have a lot of genetic diversity in your study, there's a risk that you find variants that are not really linked to disease. It's just, we call it population structure. And since studies had started to focus, the first few studies had been in people of European ancestry, it was the easiest thing to keep going and to build on those existing collections. And then it was just, a lot of the code had been written for this group. And so now, when a very small number of participants from another ancestry group were available, it was just not worth the hustle of, all this added complexity, people thought. And that's just, I think it's just spiraled into people would just always go the easiest way. The bigger picture for sure is that we're living in a society that has values and norms. If suddenly people of European ancestry weren't included in genomic research, we wouldn't, it wouldn't have been possible. It wouldn't have happened, right? So I think social values, bias, and has clearly contributed, and they impact on research at every level, right? From the politics down to the communities of researchers. If they're not representative and diverse, there's nobody, to change my mind, it needed a student who, did not identify with the majority white ethnic group to change my mind. So we need a lot of these people also in positions of influence. We need to value diversity. And that clearly hasn't happened for a very long time.
Like you say, it's definitely a systemic issue. And there was a point that you sort of picked up on there, that how does lack of diversity among researchers also kind of drive this bias?
Yeah, exactly. As I said, I think this is complex, but so first of all, demanding, if you yourself, if there's a more diverse group in research, I think the norms are going to shift, and it just won't be acceptable to continue in this current way anymore. And the other thing is that, even the research questions you ask may be slightly different. The types of diseases you focus on, may not be identical. Being from a particular group gives you a certain mindset, and we're influenced by it in all sorts of subtle ways that we don't realize, and that impacts massively on our research. There is no such thing as objective science. It doesn't exist.
You mentioned there something as well, and before that I wanted to sort of pick up on, but does this lack of representation sort of impact on research around rare variants, more so? Is that, or how does that influence things?
That's a really interesting question. You are right. There are probably some differences in the rare variant field and in the more common variant field. And I think, so there has probably been more diversity in the rare variant field because certain populations have advantages in discovering, especially recessive disorders. So groups with more consanguinity are more likely to have recessively inherited rare variants. And so there has been a longer standing tradition in doing research in communities or countries with more consanguinity. The other thing is that there is a general assumption in the rare variant field that findings would generally be more portable. But I'm not entirely sure it's always true. There's definitely, so inversely, then maybe this lack of there wasn't the same level of outcry in the research groups as there was in the common variant field, just realizing with the big numbers and the big studies how biased they were. So yeah, maybe the change hasn't quite arrived in that field as much as the common variant.
Yeah, that's brilliant. But yeah, so obviously you've talked about some of the factors that have contributed to this, but the other main focus of your sort of paper and generally your work is what we can do now. So yeah, so I guess how can we ensure that populations aren't left out in the future?
Yeah, that's a great question. So the first thing is, I would say, surprisingly simple. Lots of researchers use existing data, existing studies, and many of these studies have at least some participants who are more diverse. The most widely used genetics research is called UK Biobank. It has 500,000 participants, so it's gigantic, but it also has around 38,000 participants with non-European ancestry. And the vast majority, over 90% of studies, exclude all those participants. So a fairly simple starting point would be to not use a default analysis model where you're excluding everybody else. And methods have caught up with it, so it's not actually necessary to exclude everybody else. So that's a very simple first thing to make, and I think that more samples, better generalizability, it should benefit everybody. Equally, what you can do now, if you're a reviewer and you have a European ancestry only study, you should at least ask, why. I think we need to move on from a perspective where that's a widely acceptable approach. Then, of course, doing a study, designing a study, I don't see any reason anymore to focus it on European ancestry only. There are lots of arguments actually to possibly even focus on other groups. Sometimes it's a higher burden of disease, or actually more potential to discover new things that we didn't know about because they have been understudied. Of course, try to find collaborators who are diverse in lots of ways from you, because eventually the research is very likely to be better, and there might be questions and ways that you haven't considered before. Community engagement, and it's something that any researcher could actually do fairly easily, is to work with those communities, interact with them somehow, reach out, talk to patient representatives. Again, I think the indirect impact, even if you don't instantly have a massive study that is very diverse, but it's likely to change your mindset. And then, of course, funding. We need to look at funding, and it's important that funders value diversity as much as it's important that the research community does.
Yeah, you've covered off some pretty big topics there. I realized that was a big question, but yeah, we are dealing with something that's we've got a very wide scope. And yeah, like you say, I mean, it's not just genetics. The patient participation that you mentioned there is a massive point that we need to explore more. I would just wanted to go back because you mentioned the UK Biobank data. This is quite a tricky question for you, but I guess I was just thinking around, obviously that diverse data is there. What do you think the steps are to pushing researchers to actually using that data? Is it a question of just general education, or is it like better tools? What do you think we can do to encourage people to use that diverse data?
Yeah, better tools are definitely the one thing that would make a gigantic difference. Of course, also, and I think that is actually happening more and more, better Well, education, communication, changing the norms, discussing the value of these data. But practically, most of the time, busy research groups with different levels of expertise, they have their scripts and their way to analyze data in place. And that's been great for genomic research, is that it became easier and easier to do analyses. And it's not yet as easy. People are very unsure how to handle more diversity in data sets. And indeed, some of the more specialized software that is really good at managing diversity is really difficult to use. So absolutely very important to create pipelines that everybody knows, that are really easy to use, software. And not everybody is a sort of coder or statistician in the field. Then, lots of people with a medical background or coming from different areas. So they're not always super computer savvy. And it's great that there's lots of different backgrounds in the field. But yeah, you need an easy to use tool. I have tried to find funding to create that, but I haven't been successful so far. So for a few years, let's see. But yeah,
Yeah, I think obviously that kind of moves on nicely to us focusing a bit more on some of your work and like how you're working to fill this gap in your own research. So could you tell us a bit about that? We'll go into some of the specifics later, but yeah, how are you working to fill this gap in your own research?
in several ways. So we are working on our own tools that are helping to address some of these questions. With my group and collaborators, we're asking more fundamental questions beyond a specific disease. For example, if you do have, we have lots of findings already, are they generally transferable? Do they apply to everybody else, or do we have to be careful about that? And then we do specifically do research using existing data sets that's more diverse on a number of outcomes. The biggest for us being at the moment major depression. And the final one is doing a large study, so recruiting data ourselves and supporting others that do collect new data and do recruit. So yeah, that's the various approaches we're using.
That's brilliant. Yeah, so I guess part of that and something you mentioned previously is kind of global collaboration and initiatives. I wondered if you could tell us a bit about the H3 Africa initiative and how the work that you do that's involved with that initiative.
Yes, sure. I'm not directly a part of H3 Africa, but it is a really a massive step change. It's a gigantic collaborative effort in Africa. It's funded by Wellcome and the NIH. And it is the biggest project around genetics in different African countries. It's done in a way, it's done in a way that it's led by African investigators, which is really extremely important, rather than Western investigators, that go into African countries and take out samples. The aim is to cover a lot of the diversity across different regions in Africa. So there are a large number of study sites in different countries and investigators running projects. And the scale is really large. So I think they're targeting to get to genotyping of around 70,000 participants, which is amazing. So yeah, I think that is going to change the field to some extent. And the African continent in particular has been one of the most left out in terms of doing genetic research, although it's the area in the world where there's by far the most genetic diversity.
Yeah. And I guess the next step will just be making sure people actually use that resource. But yeah, I mean, it sounds like an amazing initiative. So we'll focus in on some of your own research now. So could you tell us a little bit about some of the key findings from your recent work into depression in diverse populations?
Yes, absolutely. Depression is an interesting disease to study because it is a mental illness. It's very, very common, and it's actually very severe. It has a gigantic burden. Some people are not aware, but it is an actual disease, and it's one of the diseases that has probably the biggest impact, on the biggest burden on the world. Depression is obviously a global disease. It impacts people everywhere, although there are some differences in prevalence. And just like with almost any other complex disease, the majority of research had so far been done in white people. Depression is also interesting because it's heritable. It's about 30 to 40% of the variation risk is genetic, but it's very complex. And that meant that the first genetic studies really struggled to find anything. So depression was, it took ages. There's been, lots of discoveries for other diseases, and it wasn't clear why things weren't going well for depression. And eventually it turned out that, it's just that there isn't one major gene involved in depression. It looks like there's loads of genes, and they just have a really small impact. Interestingly, the first study that found a gene linked to depression was done in China, in Chinese people. But after that effort, not much happened in terms of diversity. So all of the big studies, and they have now become really gigantic. The published work is more than 100,000 cases, and the next upcoming studies includes over a million people in total. So really big, really fantastic, and lots of depression genes have now been identified. Published, it's over 100, let's say genetic variants to be more precise. But in the latest release, we're talking about 500 or so. So with all of this wonderful progress, we were asking, what about people of diverse ancestry? And so we set out and looked for data, and mostly used existing data. So they were actually there. Anybody could have done this. And we just went around to other researchers, all sorts of biobanks and online resources, to get every little bit of data, any study where people included measures of depression and had some diversity. And we put all of that together. It took us 5 years until in the end we had data from 80,000 cases, so people with depression, and loads of unaffected people with diverse ancestry. And it was great. We used that for discovery. We found around 50 new variants linked to depression, which is great. And we also tested if the existing findings are transferable. And we were quite surprised to see that the picture is mixed. Many of the genes are not actually universally impacting on depression. And we don't quite understand why yet. Based on the research we've done for other diseases, things tend to be fairly transferable. There are interesting exceptions, but for depression, it just seems to be more complex. And there are lots of potential explanations, maybe, the environment, maybe depression is actually not just one disease, but, lots of different little subgroups of diseases, and maybe different in different countries and within different ethnic groups. You may be recruiting slightly different cases. We don't know, but I think it's an important reminder that you can't just an example of why it's so bad to do research in one group only. Your findings may not be generalizable.
Yeah, Yeah, exactly. That's a really good example of that. That's really interesting. To hear that it might be a group of like lots of different diseases. I'd never really thought about it like that. Um, is there sort of future work that you might do to sort of how, I guess like my question is, how would you discover that if it wasn't lots and lots of different diseases that were all being branched under depression? Um,
Yeah, really good question. So we're actually hoping that genetics could help with that. So it's extremely complex to just look at the symptoms somebody has, because they can be so diverse. Two patients may have almost no overlap in symptoms. It is a very heterogeneous disease. But if we use genetics, that's why we're hoping we could actually look at patient groups that are, share the same genetic risk factors, and that might be relevant to inform treatment.
yeah.
Which is the other complicated area in genetics, actually, that current treatment doesn't work for everybody. So hopefully genetics can shed some light on that.
That would be great. I think this kind of follows on nicely. I believe you've done like another study in Pakistan specifically on mental health disorders more broadly, so not just depression. Could you tell us a little bit about that and perhaps some of the sort of key findings from that study?
Yeah, that's right. So in this current depression paper that I mentioned, the group that's actually the smallest major ancestry group were people of South Asian ancestry. And that's true more generally. There's actually also really little research on the South Asian continent, and equally here in the UK or the US for people with South Asian ancestry. These things are about to change, but it was really important to us to make a difference there as well. So I'm collaborating with two amazing researchers from Pakistan, and also an American researcher, Jim Knowles, and in Pakistan it's Professor Mohammed Ayub, who also recently joined UCL as a site affiliation, and Dr. Aslan Hassan. And we found several sources of funding to put together this really large study of mental illnesses in Pakistan. So we're recruiting around 10,000 patients with depression, 10,000 patients with schizophrenia, and 10,000 with bipolar disorder. So 30,000 cases in total. And we're recruiting controls as well. And this study has been really, so the recruitment is happening right now. We have a few thousand for depression so far. Schizophrenia has actually been completed, the recruitment, and bipolar is ongoing as well. And it's just been an incredibly eye-opening experience to do this study.
Yeah, I can imagine. This links back to something you mentioned before, actually, which might be more specific to depression. But you mentioned that prevalence kind of differs across populations. I wonder if you could tell us a bit more about that.
Yeah, so there are some groups or countries where prevalence of depression is really high. In the UK, it's on the high end. In the US, in some other European countries, and there are other countries in the world where it's much lower. Japan, for example. It's not really well understood, and it's very, very difficult to study why that may be the case. There's also a number of countries like Pakistan where it's actually not really well known how high the prevalence is. There's been work, for example, by the WHO to try and do this big global comparisons, but they also acknowledge that the data is so weak at the moment. There haven't been enough large studies that were comparable and well conducted. So we don't really know. My hypothesis is not that these are genetic differences. I think it's much more likely that these are the immense environmental differences that impact on people's lives and increase risk of depression. And they can be quite complex. So, I mean, some of them, there may be quite a few in Western countries that, the way we live that exposes us, stress is really a well-established risk factor, for example. But in Pakistan, there's definitely an accumulation of risk factors that tend to be slightly different. For example, in certain regions, there is an ongoing problem of terrorism. So loads of people will have been exposed to, violence of some form, witnessing bomb blast, losing a loved one, really, really awful traumatic experiences. So it is clear that, on a population level, they will impact on people's lives and their mental health, and they're not equal across countries. Equally, poverty, there's poverty and things linked to it, fear of being hungry tonight or not being able to pay the rent, or having, our refugees. There are lots of refugees in Pakistan right now as well. People who've lost their homes, people who came from Afghanistan. So it's easy to imagine how such very, very difficult life circumstances increase somebody's risk of not just depression, but also other mental illnesses.
Yeah, no, that's incredibly interesting. And I think obviously the fact that it's such a complex disease as well, and that there's no sort of surefire sort of genetic test that's going to say, yes, you've got depression. I guess that makes it kind of more difficult to detect. I think you were sort of suggesting this as well, but I guess, is there an influence of it's kind of reported less in certain countries? So maybe in the UK and Western countries, we're more aware of what depression is in like a simplistic sense. I guess that would have an impact as well, perhaps.
Huge impact, absolutely. So whether somebody reports it and how it's reported, in the UK, even if you go back, so, kind of by age group, cohort, a few generations, reporting of depression was much less. So if today you look at different ages and you ask, there'll be very different likelihoods that somebody in a certain decade would say yes. People also report it differently, and there's a lot of research on that. For example, in the UK and in the US, the sort of emotional side of it, sadness and so on, that's a key symptom that is reported, and it's also important for diagnosing it. But traditionally in China, that was not a commonly reported symptom. There would be more a bigger focus on the somatic symptoms, so more of the physical symptoms, but also how these emotional changes would be described and recognized can be quite different. So it is very hard to do this internationally, but again, really important. And there probably isn't the one also correct way. Obviously, stigma is a big problem, but it's truly possible for people to have the disease and just experience it quite differently. We're also shaped by how we view the world, our life experiences, how we talk about ourselves, how we used to perceive and interpret our emotions.
Yeah, and I guess that makes it very difficult to study as well. So yeah, we've spent a bit of time on sort of the mental health disorders and that aspect of your research, but obviously you also do research in lots of other complex diseases. Just as an example, I wondered if you could tell us about some of the interesting findings about cholesterol in Uganda.
Yeah, that was an interesting earlier study we have done. So here we looked at, as you say, cholesterol levels. Most people will be quite familiar with it, right? It's such an important risk factor for cardiovascular disease. So having high cholesterol is bad. In fact, what we mostly focus on is having high LDL cholesterol. And it's such a commonly used biomarker. It's also easy to measure from blood. It's really one of the most important clinical blood biomarkers. So we didn't actually expect to see what we found. We assumed that the genetics of it would be fairly constant across the world, but we did one of the first studies with a truly global source of different data sets, including, as you mentioned, a study from Uganda. And we just compared findings both in terms of individual variants, if they impacted on cholesterol levels in different groups, as well as genetic risk prediction. And what we found was that specifically for one type of this lipid biomarkers, triglycerides, which is more like essentially it's almost like a type of fat molecule in your actual blood, there were really big differences. So the findings from big European ancestry GWAS were not transferable to the Ugandans. That was quite surprising to us. And it's hard to understand, but if I may speculate, I am suspecting that this may be related to diet. And again, something we're not usually accounting for in genetic research. The diet of those people from this Ugandan study is so different. So this is a rural Ugandan cohort. They also were younger than the other studies. In general, they had a more favorable cholesterol profile. There was very low rates of obesity. If you compare that to a UK-based general cohort of the same time, we're looking quite different. And yeah, so I suspect that some of these genes only impact on your blood lipids in the context where people eat certain things and have a certain diet. I think biologically is plausible, but we don't generally account for this in genetic research. We would like, we don't do much with the environment at all. It's more like an annoying thing that makes things more complicated. And we generally assume if we have a finding that it would just always be valid across all types of people and environments. That's clearly not always the case.
Yeah, and I think it's a very, very interesting area. But yeah, it's something we've kind of touched on in various different questions here. But those gene environment interactions, that's such an interesting area of research. I guess, what is the sort of potential role? You've mentioned there that there could be something linking cholesterol. That could have such a big impact on how we transfer those findings to different populations.
Yeah, precisely. And thinking it through, it may well be that also within the UK, there are parts of the population where certain things don't apply, or mechanisms are slightly different. Because obviously not everybody has the same diet. We just have these big studies and numbers get so large, we kind of average things out. But do we know whether our genetics findings and our genetics predictions are truly applicable to everybody? I don't think so. The challenge is that, environment is so complex and we often don't know what we should be looking for. So this gigantic search space between lots of genetic factors and lots of potential environmental factors makes it so hard to pin these complex mechanisms down.
Yeah. Do you think this is an area that will be sort of looked at more in the future? Or I guess, what are your hopes for the future in terms of that specifically? It seems like a pretty incredibly important thing to start taking account for in these studies.
Yeah, I think this is where definitely we would want to be heading. The last 15 or so years, we've largely spent on finding genetic variants in very, very simple study designs in a setup. And from the vast majority, we don't understand mechanism actually, and we don't understand who they, if they are absolutely universally applicable or not. And that's really where what we want, what we need, what the greatest benefit of genetics could be is mechanism. If you think about all of these complex diseases, for many of them, we have a very bad understanding of mechanism. Depression is one of them, but lots of others are equally complex, and even in super well-studied diseases like common cancers or cardiovascular disease, there's still a lot of question marks. So what we have to do now is, be more specific, be more diverse, and have the breadth and diversity of participants and different, people. It's not just ancestry, it's also, diversity in terms of lifestyle, age, sex, really importantly, socioeconomic background, to start looking at these things and then really try and study the mechanisms behind different genetic associations.
Yeah, that's great. And just one more area that I haven't asked you about yet. I think some of your current work is focused on sort of creating optimal analytical approaches for these, ancestrally diverse samples. So I wondered if you could tell us, I guess, a bit about the challenges you're trying to overcome. So why is it so challenging to analyze these samples? And then some of the tools that you are creating to overcome these challenges.
Yes, sure. So in general, the main challenge, I'd say, in the field is about when you're trying to find new genetic variants, it's this fear that the differences, the subtle differences even in ancestry, could result in findings that are actually false positives. So practically, it may mean that you have, say, there are, in a very simple case, two ancestry groups in your study, and they are different. They are two different ancestry groups because some variants differ in frequency between them. Now, if we study depression, and depression is also more common in one group, any variant that happens to be more common in that group looks like it leads to depression. But actually, it's just slight ancestry differences. This problem, known as population stratification, that's been the one thing, the one cause for anxiety. There are some standard methods that have been around for ages and that most people would use, such as accounting for ancestry with covariates, for example. And they work much better than expected a lot of the time. So I think the problem has been the fear is bigger than it has to be sometimes. But there are newer methods, for example, mixed models that can look at both ancestry, but also include relatives, which is really great. They are a lot more computationally demanding, and they take a lot more time to run, and they need more expertise. If these methods were more easily available, that would be fantastic. And sometimes it's just really simple thing like data cleaning. People are often unsure about how to do data cleaning when you have different ancestry groups, because it is true, some of your standard ways of doing that wouldn't apply. But there are lots of methods for lots of little questions that are being developed by us and others. Part of the challenge is that the field, there isn't really an incentive to create great tools, but make them very user-friendly and maintain them. As scientists, we don't get grants for that. You get maybe a grant to develop this method, but then it will be, you put it out there, it will be stuck, it will never be updated. You don't have time to help anybody apply it. And it becomes so hard that eventually nobody will. So there's an incentives problem definitely in the field. Genomics England is actually trying to address this to some extent. And my role there is partly to support also a tooling initiative where the group is trying to get an overview of all of the existing tools and then try to find ways to make them more accessible, nicer, and that would also help us get a better idea of what it is that's missing. Right. For many applications for many questions, there are applications. It's just not very well known. And so researchers might be unsure how they can address a certain challenge.
Well, that would be great for future research. But yeah, to end, I think, you sort of mentioned previously about some of where you think the field is going and hopefully how we will have more diverse data. But do you have any sort of hopes or wishes for future developments? Or perhaps if you could tell us where your research is going to focus on in the future?
I think one key hope is that we'll just become a lot more reflective, self-reflective as a community of researchers, self-critical, and necessarily more diverse to begin with. I think the field is it's already become extremely collaborative, but that was still very often limited to the US, the UK, maybe Australia once in a while, European countries. And that's changing. So H3 Africa is one example, but truly global initiatives with actual partnerships rather than Western researchers dominating this. It's my personal aim, and I have some fantastic collaborations, but I think more widely that's a development in the field. And then what we need to urgently do as part of this is capacity building. And that's within our own countries. I mean, in the UK, in order to be a more diverse group of researchers, we need to make sure from nursery onwards that people are equally supported, and that we end up getting that all of the super talented people from all of the different ethnic groups and backgrounds have the opportunity to do research if that's what they want to do. So that's a big problem right now. It's really fundamental. But of course, there's a global aspect to that as well. We also need to do capacity building to make sure all the talented people, for example, that I'm working with in Pakistan, who are really amazing, get access to good training. And we will all benefit from better research, better collaborations, and yeah.
Yeah, definitely. Like you say, it's not sort of just having an impact on those certain populations, it's actually impacting everyone. Yeah, that's great. That's actually all we've got time for today. I've certainly learned so much from our chat. So I would just like to say thank you again for taking the time to answer these questions with us.
Thank you so much, Lauren. It was really great, and yeah, very interesting, fantastic questions. Thank you.
Thank you.